First of all, I'd like to thank the organizers for organizing this meeting after this gap of 3 years. So today I'm going to talk about a model which would be describing the effect of contact inhibition locomotion on cellular organization in confined geometry. So contact inhibition locomotion refers to this phenomenon of cells reorienting their direction of movement when they come in contact with other cells. And this is schematically represented here, that you see that when the cells come in contact, they have this tendency to reorient away from each other. In fact, it is seen in wide class of cells like neural crest cells or Martin Derby kidney cells and other classes of cells. And it is believed that this effect or this propensity of the cells to reorient is crucial to understanding even the process of wound healing or cancer cell migration. So here are some experiments which are maybe here. It's not okay. This movie is not playing, but what I wanted to show here was that there are all these experiments which have done on this kind of fibronectin coated 1D substrate, where the cells come in contact with each other and then they are reorient away from each other. So this kind of experiments in confined geometry allows one to more quantitatively quantify the effect of CIL in such confined geometry. And today I'm going to talk about a model which mimics the effect of this CIL, or rather the interplay of this effect of contact inhibition locomotion with the cell motility in this kind of confined 1D geometry. So here is the model. So the model is a discrete lattice gas model. So we represent this fibronectin coated 1D substrate as a 1D lattice, and we treat these cells as some particles which can hop unidirectionally depending on their polarity. So the cell can have two polarities, that is, they're moving from left to right or from right to left, and then they hop unidirectionally with some rate A, provided that site on the neighboring site, the adjacent site is vacant. So we put in also excluded volume. And then the effect of CIL is mimicked by this Hamiltonian. So when the cells come in contact with each other, the effect of this contact inhibition locomotion is modeled by this Hamiltonian, where we express it in terms of some CIL parameters J1 and J2, which basically the effect is that if the two cells are facing each other, then there is some energy cost, and it is energetically favorable for the cells to be oppositely be oriented. And here theta are Heaviside theta functions. So this mimics the effect of CIL. And like I said, the translation dynamics is mimicked by this ability of the cells to hop unidirectionally in one sense with this excluded volume effect. Now, and here I'm just showing again these experiments which was done on this MDKMA cells, where one can actually see, and these time scales are typically of the order of a couple of hours. So this is like around for 2 hours, where you actually can see that the polarity of the cells, how they reorient locally when they come in contact with each other. So the first situation I consider is when you have such substrate which is completely filled by this adherent cell, and in that situation that would correspond to what is called the confluent state. And in such situations, of course, there are no vacancies in the lattice and there is no dynamics, so there is no translation dynamics. However, there would be, due to this effect of this contact inhibition locomotion, there would be dynamics related to flipping the directions of the polarities of the individual cell. But this Hamiltonian, one can use the standard techniques of transfer matrix to solve or to obtain exactly the, for instance, what would be the average energy associated with this configurations, or what is the correlation function. And similarly, one can also look at the effect of an external field which couples to this polarization. And here I just show a comparison of this average energy per cell, or the average polarization per cell with corresponding Monte Carlo simulations. One more thing I would just like to point out, there have been earlier modeling attempts which are more continuum version of the model, where the model of this CIL is in some sense symmetric, in the sense that would correspond to a special situation where this coupling strengths of J1 and J2 are exactly same. Yeah? So here in general, we would look also at the effect of this asymmetry of this CIL interaction which are modeled through this J1 and J2. So after I have discussed this confluent state behavior, now I come to the more interesting question that when you have these cells and you have vacancies, that means when you have both the motility of these individual cells and these interact and their interaction through CIL, then one parameter that we characterize is that one important quantity is a ratio of these hopping rates, the bare hopping rates and the bare switching rates, which I denote by this ratio Q. And here what I show you is the plot, the spatiotemporal evolution of a cluster. So this direction is along the lattice. The blue and the red particles just correspond to the polarity of the individual particles, and this is along the time axis. What you see here is, this is, I am showing you the steady state configurations. As you increase Q, that means if you increase the ratio of this hopping to switching rate, you have a tendency of these cluster size being larger, which you can clearly see. You can also, what you also notice is that as you go to this high Q regime, you would have typically clusters where the boundaries of the clusters would have these plus particles or the right moving particles on the left end of the clusters, and on the other hand, on the right end of the cluster, you will have particles which have left polarities, that is particles moving to the left. Now, next question is that we look at the cluster size distribution of these particles in different regimes. So, in the regime where the switching rate of these particles is much faster than the hopping rate, there actually this model will get reduced, so it would be identical to the results of a totally asymmetric exclusion process or symmetric exclusion process. And it is well known that the cluster size distributions are exponential, rho are the particle densities, etc. So this is in this regime, and indeed the Monte Carlo simulations bear that out. So these plots correspond to this this limit of Q being much lesser than 1. On the other hand, as I go to this high Q regime where the switching rate are relatively much smaller than the hopping rate, then what you notice is that, of course, the average cluster size increases, but what you also notice is that the tails of the distribution still continue to be exponentially distributed, which are shown by this similar plot. And here now I come to these diagrams or the contour maps which characterize this is the overall behavior in the system. Here the two axes are the Q, which I just mentioned, and here you have these parameters which characterize the CIL strength. In the first contour map, I look at the case where this J1 and J2 are symmetric, and then the effect that we were mentioning, that you of course expect that when you increase or rather when you decrease the switching rate, then you have a propensity for larger clusters to stabilize. So if you go along this axis, in general, the average size of the clusters would increase. On the other hand, the effect of CIL is that CIL in general likes the clusters to be of smaller size. In fact, that is what is captured here, that if you go along this axis, the cluster size actually decreases, the average cluster size decreases. However, if we look at the asymmetric case where this J1 and J2 parameters are different, so we look at the case where we hold some fixed value of J2 and increase J1, then you notice that there is this interesting behavior. Of course, if you are below a certain threshold value of Q, then what you notice is that as you increase this J1, the cluster size decreases. However, for certain, if it's above certain threshold value of Q, above a certain, you have this kind of reentrant behavior. So if I go along this axis, along the J1 axis, what you notice is that initially the cluster size decreases. However, if at some point it will again actually, the cluster size actually starts increasing. These are some of, so here I have plotted the average cluster size as a function of J1, and you notice this kind of reentrant behavior, which is exhibited and which is arising out of this asymmetric nature of the CIL interaction, which can lead to this kind of behavior. We also observe in the simulation that this average actually, so the average cluster size is monotonically increasing function of Q. And another observation was that if you look at the average cluster size in this high Q regime, then it scales as, it goes as Q to the power of half. So how does that dependence come about? So for that, one can actually, in the high Q regime, one can map this model to an equivalent equilibrium process. So in this high Q regime, typically the situation would be you will have these different clusters, and once in a while whenever a particle at the cluster edges flip their directions, they will quickly join the next cluster. And in this limit, you can think of the evolution of these cluster dynamics to be independent of each other. And then one can talk in terms of the configurational entropy that is associated with different cluster sizes. And one can associate a configurational entropy, and of course there is an average energy associated with the cluster, and you can get the cluster size distribution in this limit approximately by minimizing an effective Helmholtz free energy. And once you do that, 2 more minutes. Once you do that, okay, some of the things are missing, but basically you at least approximately see this kind of q to the, not very nicely, but at least approximately you see this kind of half power dependence on q. These are the comparison of this analytics calculation with the simulation result. And finally, one can also look at what are the polarization characteristics within a cluster that there would be in general. If it were, if no CIL was there, you would expect this kind of associated with a binomial distribution m to the cluster. It will go as just minus half power cluster size. Otherwise, there would be a polarization. I'll sort of skip what would be the effect of the field and come to the conclusion. So what we have shown here is that you have a confluent state where you can have an exactly solvable model, and how the average cluster size depends non-monotonically on the CIL strength, if there is, especially if there is an asymmetry in the CIL interaction. And then the corresponding contour plots exhibits this kind of reentrant-like behavior in this space. What we do not observe is this kind of motility-induced phase separation, at least for this model. There have been a lot of debates on that. What we plan to do, or what needs to be incorporated, or what I've neglected here, is the interplay of the contact inhibition locomotion with even cell-cell adhesion, which we have not taken into account, and how that will manifest in this kind of organizations in 1D, and finally extend this kind of model to generalize it to 2D and compare it with more continuum hydrodynamic model. So with that, I would come to the conclusion. Here are the co-workers. Herschel is a PhD student at SPPU, and this was jointly done with Ignacio, who is at University of Barcelona. Thank you for your attention.